Hello again and welcome to part 4 of this 8-part series looking at a beginner's guide to colour genetics in budgerigars. In the last 3 parts, or in the first 3 parts of this series, we looked at the way a budgerigar inherits various varieties and mutations. We looked at the genetic makeup, so how the mutated allele interacts with each other, and we found out these could be either dominant, recessive or co-dominant. In fact, in the last video we covered co-dominant. So we now understand how a various variety or a mutation can be passed on from one generation to the next based on those inheritance factors. We also learnt that a bird can be either single or double factor, and in order to help us predict outcomes of pairing 2 birds together, we used the Punnett Square. But up until now, all we've done is we've looked at a single gene when we've been using the Punnett Square. Well, as we all know, budgerigars don't come in quite such easy to follow. So most budgerigars have more than one variety. So we can have, a blue spangle or a yellow face dominant pied, for example. So how do we use the Punnett Square when we're trying to predict more than one variety or more than one gene type on our birds? And that's what part 4 is going to look at. Before we do though, as always, don't forget, if you do enjoy these videos, then do hit the like button and of course subscribe. And don't forget that over on the Shed Talk Facebook page there is a PDF booklet that you can use when you're following this guide, and it gives most of all the detail that's in the video guide, but in a little bit more detail. So do go across and join the Shed Talk Facebook page, and you can find the PDF document there. It's free to download, so there's no cost involved, and I know a number of people have already done that. So let's now move on then and consider predicting multiple genes. So as I just said, it's not always that simple when we're considering pairing 2 of our birds together, that the chances of them just having one mutation are likely to be fairly rare. So we're going to consider now how we predict or take forward that and start predicting multiple genes. And for this particular example, we're going to look at a sky blue single factor spangle and a single factor grey-green split for blue. Now if any of those terms you don't understand, then do go back and just review the first 3 parts of this series, because you should understand what I mean by single factor and you should understand what I mean by split for blue. So we're going to consider a sky blue single factor spangle and a single factor grey-green split for blue. Now I know that the single factor and double factor grey, because the grey gene is dominant, I know that there's no difference really between a single factor and a double factor grey-green. However, from the point of view of working out the inheritance, it is important whether we know whether the bird is a single factor or a double factor for the grey allele or the mutated grey allele. So that's what we're going to look at, a sky blue single factor spangle and a single factor grey-green split for blue. So because we're looking at the more than one gene for each of the birds, it does make the process slightly more complicated than the way we have looked at so far. It's not overly complicated, but to make it easier though, I tend to break it down into 5 phases, and that's what I'm going to run through now. And the first of those phases is to try and identify which genes each of the bird has, so we understand what genes we need to focus on and what allele we need to focus on for the Punnett Square. So if we take, for example, the birds we were just looking at, the sky blue single factor spangle, we know, because it's a sky blue and because it's blue and the blue allele is recessive, that it must contain both of the, or two allele for the recessive type blue gene. So we also know that it's a single factor spangle, and as a single factor spangle it must contain a single mutated allele for the spangle variety. So those are the genes that we would need to look at for that particular bird, that sky blue single factor spangle. We will need to look at the blue gene, and we'll need to look at the spangle gene. Now what about the single factor grey-green split blue? Well, we know that it's split for blue, so it must contain at least one, or sorry, it must contain one of the mutated blue allele. So once again, we will need to look at the blue gene for this bird. We also know that it's a grey-green, so therefore it must contain and it's a single factor grey-green, so it must contain one of the dominant grey mutated allele. So those are the genes that we're going to look at for each of the birds, but quite clearly what we will need to do is to consider those range of genes, or those range of allele for both of the birds completely. So we'll need to consider the blue allele for both birds, or the blue gene for both birds, the spangle gene for both birds, and the grey gene for both birds, even though not both of the birds, or not each of the birds, contains all will have a mutated gene for all of those varieties. So let's have a look at what that looks like. So this is what I consider to be phase 2. I take those genes and I put them into a small table, and as you can see here, the table I've got first of all shows gene and variety along the top, and then we've got the sky blue single factor spangle and the grey-green split for blue bird, and down the left hand side I've got the various genes, so the blue and the spangle and the grey, and then the next squares represent the allele that I have identified each of those birds had. And we went through this earlier, so you can see that the sky blue single factor spangle contains two of the mutated blue allele, one mutated spangle allele, and no mutated, so both wild types, grey allele for the grey gene. Then we look at the grey-green bird that's split for blue. As I said, we know it contains one of the mutated recessive blue allele, none of the mutated spangle allele, so it contains two of the wild type spangle allele, and it contains one of the mutated dominant grey allele. So that's phase 2, is just putting those into a nice little table so I have a clear understanding of what it is I'm looking at. Once I've done that, I can then have a look at this and check each bird and see what possible combinations of those 3 genes it can pass over, and that is phase 3 of the process. So having produced the table that you can see here, what we now need to do is to work out the possible combinations that each of the 2 birds can pass on. So this is phase 3. For the sky blue single factor spangle, in effect the only different allele that he can pass on is the spangle allele, because for the blue allele, both of the allele are homozygous for the mutated gene, and for the grey allele, both the allele are the homozygous for the wild type grey gene. So in effect he's only going to pass those 2 on in the same way. For the spangle gene, you can either pass on the non-mutated wild type allele or the mutated spangle allele, the mutated allele for the spangle. So in effect, the possible combinations for the blue single factor spangle are the mutated BL1 gene, mutated blue gene, the non-mutated wild type spangle, and the non-mutated grey allele, or he can pass on the mutated blue allele, the mutated spangle allele, and the non-mutated grey allele. So those are the possible combinations. So there are only two possible combinations that he can pass on to his offspring. Let's now consider the grey-green split for blue, or single factor grey-green split for blue. So for this bird, things are slightly different because it can pass on for the blue allele, it can pass on either the non-mutated blue allele or the mutated blue allele. So there are two possible combinations there. For the spangle, the bird only contains, both alleles are homozygous for the non-mutated wild spangle gene, so therefore he's only going to pass on the non-mutated gene. But again, for the grey gene, he can pass on either the mutated grey allele or the non-mutated wild type allele for that gene. So this means in effect that he can pass on either non-mutated blue, non-mutated spangle, non-mutated grey, or he can pass on mutated blue gene, the non-mutated spangle, non-mutated grey gene, or he can pass on the the non-mutated blue gene, the non-mutated spangle but the mutated grey gene, or he can pass on the mutated blue gene, the non-mutated spangle, and the mutated grey gene. So for this bird, there are four possible combinations of the various genes that we're looking at that he can pass on. So having done that, having established those possible combinations, we now transfer those onto the Punnett square so we can work out the possible combination of offspring. I hope that makes sense. If not, just rewind and go back through it, and of course do read the various bit of chapter 4 of the guide alongside this to help you better understand it. Basically all we're doing is trying to work out what are the possible combinations of each of the gene are. So just run through them using the table and write them down as I've done here. Just use a scrap bit of paper, write down the possible combinations and just double check that you've identified all of those possible combinations. So let's now move over to the computer and have a look at the Punnett square that we will create for this pairing. So here we are over at the Punnett square, and you should remember this from previous videos and how to use it. You can see we've got the sky blue single factor spangle across the top, and the two possible ways that they can pass on their combinations of allele for each of the three genes we're considering. And on the left hand side we've got the single factor grey-green split for blue, and we've got the four possible combinations of allele that bird can pass on for the three genes that we are considering. So exactly the same as we've done previously, we just need to pull the allele from the top bird for each of the squares down, and then add the allele from the left, the bird on the left hand side, bearing in mind this time that we've got three genes to consider. So I'll just do the first. So there you go, I've completed the first square, and you can see we've brought the BL1 gene down, or allele down, and the B across, the small s, so the non-mutated spangle allele from the top bird down, and the same across from from the left-hand bird, and the same for the non-mutated grey allele for each of the birds. So that's how you complete it. Fairly straightforward. What you can do now is if you pause the video now and just have a go at completing the rest of it, and then when we come back we will have the completed square and you can compare yours with mine, and then we'll talk about what each of those various outcomes might be. So if you pause the video now. So here we are. This is the outcomes that I've come up with. As you can see, there are a total of 8 possible outcomes, and in this case they're all different. That might not always be the case. Sometimes you might get a couple that are the same, but in this case all 8 options are different. So from these 2 birds, the possible outcomes are 8 different mix of varieties. What we need to do now is to work out what each of those varieties are. So for example, if we start with the top left hand one here, the one we did originally, we can see that this bird contains one of the mutated blue genes and one of the wild type blue genes, and we know that the mutated blue gene is recessive, so this is going to be a green split for blue bird, and it contains none of the mutated genes for either the spangle or the grey. So in this case this is going to be a normal light green bird. So from the grey-green and the spangle bird we've actually got a normal light green bird. And so if we look at the one across here, the next one across, we can see that this is also a normal light green split for blue, but this time it's a normal light green single factor spangle split for blue. So there's another possible outcome. See if you can work out the remainder of these, and then what we'll do is we will then have a look at all of the outcomes that I've identified with this and do a comparison. So once again, if you want to pause the video, you can have a go at completing that, working out what each of those birds might varieties might be, or mix of varieties might be, and then when we come back we can do the comparison. Okay, so we're now on to phase 5, as I said, looking at what the possible, the various possible outcomes of the birds might be, and I've written them down here, and as you can see I've put the percentage possible outcome for each of these. So each of them there is a 12.5% possibility of one of the birds producing this, and from each of the various possible outcomes we can see we've ended up with a single factor, or 12.5% of the birds are likely to be single factor grey-green, single factor spangle split for blue. Then another 12.5% are going to be single factor grey-green split for blue. 12.5% are going to be single factor grey, single factor spangles. And they're all 12.5%, so we've then got just a single factor grey, a light green single factor spangle split for blue, light green split for blue, a sky blue single factor spangle, and a sky blue. So from those 2 birds, 8 possible outcomes we could have, from sky blue through to what looks like a normal light green but is actually light green split for blue, through to a grey birds and a mix of spangle and grey greens. So there you go. I hope you enjoyed that little example, and you can see that it's not particularly difficult to work them out, and you can get some quite complex results from it. As I say, it's the easiest way to do it from my point of view is just to split it down into those 5 steps, work through it in that logical way, a logical process from one end to the other, and you should be able to come out with it. As I said before, if you need to look at this in a bit more depth, then do go across and consider looking at the PDF document which goes through it and gives a number of other possible examples of or a number of other examples that you can try out. try it with some of your birds. Have a look at what your birds are, guess roughly what their genotype is, and try mixing and matching and seeing what the possible outcomes might be. Of course, these are only possible outcomes. It might be that you end up with all the same birds in the nest, because it's just the chances of it happening are as I say, the percentages that we've got there, the 12.5%. So we've looked at now so far in this series how to use the Punnett Square, look at understanding the dominant, recessive and codominant varieties. What we're going to start to do now is consider what it means in terms of where the gene sits within the chromosome. So far we've ignored the chromosome completely because it hasn't really mattered with what we've been doing. But for some of the allele, it does matter which chromosome they sit on, and the first of those we're going to look at in the next video is the sex chromosome, and we'll consider those genes that are sex-linked, or so-called sex-linked. But until then, hope you enjoy the video. As always, please do subscribe and hit the like button.